everyone, so I am Shruti Oja and today we will start with cell cycle and cell division. So here I will talk about just the important points from which the questions are expected, or in with that points where you get confused mostly. So let's start with the phases, that is the interphase and the M phase. As you know, interphase is the major part and the M phase is the small part or the 5%. Okay. So interphase has three. Just remember G1, S phase and G2. So it is a G1, S and G2. And in the G1 phase and G2 phase, these are the growth phases. G stands for growth and S for synthesis. If synthesis is taking place, then CHD. Just remember childhood, CHD. So interphase is a childhood phase, right? And M phase is an adult one where the cell really divides and gives birth to new cells. So childhood, CHD, centriole, duplication or replication, DNA duplication and histone protein synthesis. This is a trick, right? I hope you know what is a trick here. So in the M phase, I am having first of all the PMAT. So how to remember in series? PMAT. Okay. And then I am having the cytokinesis. First of all the DNA will split and then the cell will split, okay, or divide. So prophase. In prophase you can see CDS. So how to remember this word? If you want a mnemonic and then you can see like see the scene. See the scene. So CDS. And why see the scene? Because it is the first phase of the division. First we have to look over everything, like we have to look over like the structure, like appearance and all, and everything is ready or not, right? You can relate it with that also. So if you need a trick, otherwise it is very easy. CDS. CDS stands for chromatin. The chromatin condense here, okay. Then centriole movement. Then disintegration of nuclear membrane and disappearance of cellular organelles, and S for spindle fiber formation. So here we are seeing the scene and preparing also. So early prophase, late prophase. In early all these events, mark my words, begin, and in late they finish or end. But here you can see disintegration of nuclear membrane. It does not finish here. It ends in the metaphase. Okay. So see, D is in middle, right? And this is the first one. So you can see the middle one ends in the metaphase. That also starts with M. So now metaphase. Metaphase, in metaphase you can see the morphology of chromosomes. M. You can relate. I have written M, M and M. Morphology, and all the chromosomes are set at the equator, middle. Mota phase. Mota is healthy, fat. So it is fully condensed. If the chromatin is not condensed, it is loosely in fibers and all. But when it is condensed, it is becoming fat. Okay, mota. Then anaphase. A for ana and alag alag. So the spindles, the chromatin, the sister chromatids are going towards the poles and they are वो अलग अलग हो रहे हैं. Then telophase. T for telophase and T for 2 nucleus. So here we will get the 2 nucleus. How? As this is the prophase is the first phase and telophase is the fourth phase. So I hope you can see CDS is also here and CDS is also here. So whatever is happening in prophase will just opposite to telophase and vice versa. So see, chromatin condense, chromatin, there will be recondensation, we can say, and centriole movement is not here because centrioles has no role after this. So it will not be here as I have written. Then disintegration of nuclear membrane, reintegration, it will integrate again, okay. And disappearance, reappearance of cell organelles. Then spindle fiber formation, and here will be spindle fiber breakdown. Next is cytokinesis. After all this has taken, we have two nucleus. Now we want the two nucleus should be different cell, right? Then cytokinesis. In animals and plants, it is different. So, just remember that animals like us are self-centered. We are more concerned about our own needs. We are taking everything from the environment but not giving to it, right? I hope you can relate it well. Not apply to everyone, but yes, it is a common fact that we are self-centered, we are heterotrophic and just taking the nutrients from the environment. So, here will be a centripetal movement. And in plants are very nice, right? They are giving everything to the environment. They are providing their services free of cost. So, it is centrifugal movement, right? And here it will be a furrow. Furrow is centripetal and centrifugal is cell plate formation. Next is here it is the microfilaments, and in plants we are having microtubules. Remember, microtubules are not present in animals. So then how will they participate now? So microfilaments and microtubules. I hope you have understood it well. Now let's move on to the meiosis in the second part of the video.